In the following, we would like to present our recent publication entitled Impact of Visual Callosal Pathway is Dependent upon Ipsilateral Thalamus. Did you ever ask yourself why you see only one image although you have two eyes? Our study answers yet unknown key details of this question. It gives you insights into the visual cortex as a gateway with many paths. You can look forward to new facts on visual perception and pathways in the brain. In mammals and many other animals, sensory information from the left side of the body is sent to the right hemisphere, and sensory information on the right side to the left hemisphere. The brain then combines the information from the two hemispheres to form a coherent representation of the whole body, and this, in part at least, involves transmission of information from left to right and from right to left hemisphere. This so-called crossing the midline principle also occurs in the visual system, although here the anatomical pathways are a little more complex. The visual cortex of the brain is that part of the cerebral cortex which processes visual information. It is located in the occipital lobe, the hindmost part of the cerebrum. Visual nerves run straight from the eye to the primary visual cortex. If we consider for a moment only the right eye, the majority of the so-called ganglion cells in the retina run across to the left visual thalamus, which is the first brain region after the eye involved in visual processing. From there, the information is transmitted to the left visual cortex. The remaining ganglion cells do not obey the general crossing the midline rule, and instead run to the right visual thalamus, and from there to the right visual cortex. This complex and highly interconnected visual system includes a large and characteristic nerve tract connecting the left and right visual cortices. These neurons are termed callosal projection neurons, after the nerve tract through which the nerve cell processes travel. The visual cortex is topographically organized based on the visual space encoded by each eye. Only the area that does represent the space directly in front of the animal has this reciprocal projection. It was suggested that the callosal pathway had the function of joining the representations of these overlapping regions in the left and right hemispheres and was critical for binocular processing and stereopsis. However, other studies provided evidence to the contrary, showing that the callosal projection contributes to, but is not essential for stereopsis. Since very few studies have investigated the role that this pathway plays in visual processing, its role remains largely unclear. So far, there have been no studies examining the functional contributions of the callosal projections to visual processing with single-cell resolution. In this study, we have investigated this in rodents. The questions we set out to address were: can the callosal projection independently drive activity in neurons in contralateral visual cortex in the rodent, and how does this pathway contribute to visual cortex processing of visual inputs? The proposed role for the callosal pathway is that it provides visual information that was sent from the right eye to the left hemisphere back to the right hemisphere. We show that the callosal pathway also transmits information from the following pathway: left eye, left visual thalamus, callosal pathway to right hemisphere. In addition, we demonstrate that though the main callosal pathway from right eye does not involve the right visual thalamus, if we block activity in the right visual thalamus, this main callosal pathway is not capable of activating neurons in the right hemisphere, meaning the right visual thalamus and right callosal pathway presumably act in a coordinated manner. In this study, we have demonstrated that visual responses in the subpopulation of binocular neurons in rodent visual cortex depend both on input from callosal projection neurons in the contralateral visual cortex and on input from the ipsilateral thalamus. In addition, we also show that callosal projection neurons do not respond differentially to visual stimuli, and that the projection targets matching regions in the contralateral visual cortex. Callosal projection neurons apparently act cooperatively with the projection from the visual thalamus, a requirement that until now was completely unknown. This suggests a much greater role of the rodent callosal pathway in cortical processing than previously thought.